Yeah.
Okay, perfect. Okay, so just to give a couple quick announcements before we get started, what I want to kind of remind you guys of is that this week we'll have our final homework assignment. And so the final homework assignment will be for chapter 11. So that will be, I think it's somewhere 14. And so that will be due on Tuesday, just like all of our normal homework assignments have been. So it'll be due on Tuesday. That's going to cover chapter 11. That'll be the last homework assignment you guys have to do for the semester. Next week, we'll have our final lab. And then, I'm trying to think what else we'll have. We'll have our classworks like normal. And those will be the last two classworks for the semester. Next week, we'll cover chapter 19. And then our final exam will be on, what is that, February 19th, or sorry, May 19th. Yeah, I'm all over the place. May 19th. So for the final, it's going to be 75 questions. The first 40 will be from chapter 11 and 19. The last 35 will be chapter numbers 41 through 75 will be from the previous exams. So between today and tomorrow, I'm going to be uploading Word document copies of the exams, so you can use those to study for the finals. And then, yeah, that's pretty much going to be it. Trying to think. Yeah, is there any questions on what's going to be going on for the rest of the semester? No? Okay. All right, so that's going to be our game plan going forward. And like I said, so we got about a week and a half left. And then if you have any questions, feel free to let me know. And then your current grades on Canvas should be pretty indicative of kind of where you are in the class. There's a few things I still have to grade, but I think I'm pretty much all the way caught up, or almost all the way caught up. So whatever grades you have on Canvas, that's pretty indicative of kind of where you're at before we head into next week and into finals. Okay. So yeah, that's pretty much that. Okay, let me go ahead and go for it here. Okay, let's clear all this out. Okay, so if there's no questions, we'll go ahead and jump into things here. So on Tuesday, we finished up by talking about the different aspects of how evolution can take place. And so we left off talking about genetic drift. So if we kind of go back a little bit, initially we were talking about genetic drift and how genetic drift is just the effect of random chance on allele frequencies. Okay? Now that being said, this random chance has a larger effect on populations that are small. Okay? So for example, we were looking at these frogs here. We had a population of 10 frogs. We lost 3 of them. And so we ended up having going from 30% brown frogs to 0%, and then 70% green frogs to now 100%. So it's a pretty large jump in our allele frequencies, and that's due to genetic drift working on a small population. Okay. If you work on a large population, you look at 100 frogs, for example, still have the same proportions, 30% to 70%. If you lose 3 brown frogs again, now you only go from 30% to 28% brown frogs versus 70 to 72%. So in larger populations, genetic drift has a much lesser effect than it does in smaller populations. In smaller populations, genetic drift has a much more profound effect. Okay. And so what happens here when we look at small populations is we'll typically end up seeing some alleles becoming fixed and then other alleles becoming eliminated. Okay. Fixed again meaning that those alleles are present in all individuals in the population, and then eliminating meaning it gets completely removed from the gene pool altogether. Okay. And so that's what we see in a small population. In a large population, allele frequencies don't end up getting fixed or eliminated because there's so many individuals in the population. And so there's a lot of random mating and things end up working out. Okay. So that's kind of where we were at. So today we're going to kind of finish up talking about genetic drift. And so one specific type of genetic drift is what's called a bottleneck effect or a bottleneck event. Okay. And so bottlenecks are either naturally or human-caused events that kill a very large portion of the population. And so when that happens, you end up wiping out a large portion of the genome because you lost so many individuals from the initial population. So for example, here, this is kind of a general schematic that has to do with, that kind of helps to explain bottlenecks. Here in our original population, we have a lot of individuals, a lot of different types of alleles that are present. You have some ginormous, kind of huge bottleneck event, whether it's kind of a volcano eruption or a hurricane or a forest fire. Something happens that ends up killing a large portion of your population. Okay? And so the individuals that survive have a much more reduced genetic diversity than the original population. Okay? And so the survivors end up having a much different genetic structure than the pre-existing population. And so what that tells us here is that our survivors, or the genome of the survivors, becomes the entire genome of the resulting population. Okay? And so again, you can see the genetic diversity becomes significantly reduced, post-disaster than it did pre-disaster. Okay. So that is genetic drift. And so when we look at bottlenecks, bottleneck events will typically kill large portions of the population for reasons other than genetics, okay, other than the genetic traits you have. Okay, so for example, a volcano eruption isn't going to kill specific deer in a population depending on the specific genes that are around, right? Volcano eruptions are going to kill just based off of whoever's around. So genes and genetic traits don't have any impact on the survivors of genetic drift events or bottleneck events. These events just kill for large portion of the population just based off of who's kind of around and who's vulnerable. So again, those are some of the natural caused events. There's also some human caused events like poaching, kind of overhunting and overfishing and things like that. So those are all kind of human caused bottleneck events that also contribute to reducing the genetic diversity found in different populations by significantly cutting down the population size. Okay. So an example of this actually happened with my spirit animal, the cheetahs. And so cheetahs underwent a pretty large bottleneck event about 10,000 years ago when they survived an ice age. Okay? And so the ice age around 10,000 years ago actually killed a whole large portion of the cheetahs that were currently present. Okay? And so there's only a few survivors that were able to make it out of the ice age. And so because there were just a few survivors, those few survivors ended up mating with one another, producing some offspring. But the offspring were all related to one another because there wasn't that much diversity in the parents. Okay? And so that reduced the genetic diversity in the cheetahs. Okay? Now, in addition to that, cheetahs also went another kind of bottleneck event that was more human caused around the last 100 to 200 years that occurred as a result of poaching. Okay? And so there was a lot of hunters, a lot of poaching that was going on to kind of gather some of the cheetah fur. Okay? And that poaching kind of reduced the cheetah population significantly to the point where they almost became extinct for a little while. Okay? And so again, that reduced genetic diversity because again, you had a very small population of cheetahs, okay, to the point where they became vulnerable to extinction. Okay? So in cheetahs, there's a very, very small genetic diversity. And to kind of put this into perspective, we'll take a little detour here. And so if you think about, let's say you needed to get a kidney transplant, right? If you need a kidney transplant, you might ask a sibling for a kidney, right? So if you ask your brother for a kidney, they give you a kidney. You're still going to need to take a lot of immunosuppressant drugs to kind of tone down your immune system so you don't reject that kidney. And the reason for that is because even though you guys are siblings, you're still different enough genetically. There's still enough genetic diversity between the two of you that your body recognizes, hey, there's something in here that wasn't here before. This is something that's not mine. Okay? Something in that kidney's genes is not of me. Okay? And so naturally, your body would have to produce a lot of antibodies to try to reject that kidney. Okay? So you take a lot of immunosuppressant drugs to tone down your immune system so you don't reject the kidney. Okay? That's what would normally happen. Okay? Now, if you do the same thing with cheetahs, let's say a cheetah needs a kidney transplant and you take that kidney and you you kind of donate it to another cheetah. If the cheetah is not going to need to take any immunosuppressant drugs because they're so closely genetic or so closely genetically similar. Okay? Even if you take two completely unrelated cheetahs, okay, they're still so close genetically that their bodies are not going to be able to recognize that there's something in there that wasn't there before. if there's something that kidney that it just received isn't its own. Okay? And so it wouldn't be able to recognize it. It wouldn't need to take any immunosuppressive drugs. And so its body would just kind of go on kind of acting as if it had two kidneys that, are, of its own. Okay? So does that kind of make sense? That's kind of to put it in perspective of how closely related all cheetahs are to one another. Okay? So in order to kind of combat that, what cheetahs will do is the female cheetahs, when they mate, they will never, or they'll almost never mate with males that are inside their own kind of general area. Okay, they'll usually travel very, very far distances in order to find males, and so in doing that, that helps them ensure that different litters that they have are never going to be closely related to any fathers that may be present. Okay, so for example, if you got a cheetah in Gilroy, for example, it might travel to Los Angeles to go mate with a cheetah, and then come back, and then have his litter. Okay, and so that ensures that the offspring and that the cheetahs themselves, the female and the male, are not closely related. Okay. And then in doing so, if they have another litter, they'll travel to, instead of going to Los Angeles, they might travel to Sacramento to go mate. And so again, that helps them to kind of ensure that the fathers of the two litters and the litters themselves are never very closely related to one another. Okay. So that's number one. And then number two, like I said, females will never return to the same males. So they never return to the same males. And again, that helps to increase the genetic diversity, because now you're creating litters with multiple different males with different genetics. Okay. And so the ultimate kind of goal of doing this is to improve the genetic variation between all the cubs. Okay. So my cheetahs are all hunky-dory, or not all hunky-dory, but they're in a much better situation than they were years and years ago. Okay. So that is genetic drift with cheetahs, or bottlenecks events. Okay. Any questions on bottlenecks? Okay, this is making sense. Okay, so from here, okay, so there's another type of genetic drift, which is called the founder effect. Okay, and so in the founder effect, typically what we end up seeing is we see a large population of individuals, and we get a subset of those individuals that leave that original population to go start a new population somewhere else, okay, at a new location. Or they get divided by some physical barrier, but they basically will start a new population somewhere else. Okay? And so they become the founders of that new population. Okay? Now, the thing about a founder effect is that the individuals that leave, okay, the founders, are unlikely to be representative of the original population. Okay? They're most likely going to be genetically different than the original population was. Okay? And so in doing so, the second thing that happens here is that the offspring that the founders kind of the founders kind of produce are more closely related to the founders themselves than the original population of the founders left. And again, that makes sense because the offspring are coming from this small subset of individuals here. Okay. So that's the founder effect. That's where you have individuals that leave a larger population to go start a new population somewhere else. And so again, that reduces the genetic diversity because again, you're working with a small set of individuals that will then begin to produce their own offspring. Okay. So an example of this happening here was with the Dutch settlers in Africa. So there are some Dutch settlers that moved from the Netherlands down to South Africa a number of, I'd probably say maybe about 100 years ago or so. And they formed what's called the Afrikaner population. So keep in mind, there's already a population of people in South Africa in that particular region, but then you also have these Dutch settlers also coming in to start the new population. And so the Afrikaners would start to mate with one another. They start to produce some offspring. But then what they started to notice is that the offspring of individuals from the Afrikaner population started to have a lot of mutations that were different than the original African populations that were already present. And so the mutations that were actually developing were actually more characteristic of the Dutch settlers, and kind of less so than the African populations. And some of the mutations that people found in the Afrikaner population were like Huntington's disease, Fanconi, anemia, and specific cancers that again were more characteristic of the Dutch settlers than the original African population. Okay. And so this is kind of an example of the founder effect, where we see founders coming from one area to another to start a new population, and they start to develop the same, the offspring starts to develop similar traits as the original founders. Okay, so that is the founder effect. Does the founder effect make sense here? Okay, cool. All right, so that's the genetic drift. The last aspect of, or the last kind of factor that can lead to evolution is called gene flow. This is just another way of saying migration. Okay, and so in gene flow, what ends up happening is we have a flow of alleles going from one population into another. Okay, so for example, here we have population 1 and population 2. And so we have some individuals that leave population 1, okay, and then bring their alleles into population 2. Okay, and in doing so, that changes the allele frequency. And as you change the allele frequency, that is ultimately going to lead to changes in the genotype frequency, the changes in the phenotype frequency, and ultimately help us observe evolution. Okay. So if we were to look at how this will take place in some deer. Here you have a western deer population. Here we have an eastern deer population and a pass in between them in a mountain range. Okay, so if we have some western deer, which have their own set of alleles, and some of them start to migrate into the eastern deer population, now we're going to be introducing new alleles into this population that is going to change the allele frequency and then ultimately show us that evolution has taken place, okay, over a period of time. And so this is basically migration. This is, again, another word for that is gene flow. Okay? So that can be either due to the flow of alleles or the flow of new gametes from one area to another. Okay? So different populations are going to go through gene flow in different manners. So here we see, how it works in animals if we're just going from population 1 to population 2. However, in plants, for example, this is going to be done a little bit differently. Okay? So in order for gene flow to take place in plants, they're usually going to need to spread their pollen, and that pollen is going to have kind of the gametes or the sperm or the sperm that's going to be of the flower. Okay? So in order for gene flow to happen in plants, because the plants cannot move themselves, they're usually going to be using pollinators. Okay? Either the wind, or they can use different animals or insects that are going to act as pollinators. Okay? And so, for example, here we have a bee, and so the bee kind of goes into a flower, picks up some of that pollen, which again contains the gametes, and then flows to a new population of plants and drops off that pollen there. Okay, and so that's going to be a way that you can introduce new alleles just by way of spreading the gametes from one population into another. Okay? And then again, in doing so, you change the allele frequencies of that new population, and then ultimately, over a period of time, that will lead to evolution occurring. Okay, that is gene flow. Any questions on gene flow or migration? Okay. All righty. So if not, we'll go ahead and finish up chapter 11 here. And so chapter 11, or sorry, 11.3, we're going to do 11.3, 11.4, and 11.5. And so what we're going to be doing today is we're going to be looking at the different ways of being able to view evolution and how we know that evolution is actually occurring. We can see some evidence of it. We're going to look at how speciation is going to take place and how we can produce new species. And then the last thing we're going to do is we're going to look at some common misconceptions about evolution. Okay, okay, so is everyone able to see this new PowerPoint? Starting with chapter 11.3. Okay. All righty, cool. Okay. So we're going to be starting with the evidence of evolution. So how do we know that evolution has taken place? Okay, so far we've looked at the different kind of causes that can lead to evolution, right? We looked at natural selection, we looked at genetic drift, we looked at mutations, we looked at gene flow, and then we also looked at non-random mating and a little bit of matters as well. Okay? So those are some of the things that can lead to evolution. But how do we know that evolution is actually taking place? Well, what we can do is we can actually look at different aspects of kind of life and be able to give a little bit of evidence and be able to observe a little bit of evidence to know that evolution is actually occurring. Okay. So the first thing that we're going to be doing, the first thing that we're going to be doing to help us identify that evolution has occurred is to look at fossils. Okay. So fossils are the remains of kind of old pre-existing organisms from years and years past. that can provide a little bit of evidence that they may have been related to a current or later species. Okay. And so those fossils will kind of give an indication of the structure or the anatomy of a previous organ or previous species that has since gone extinct, and we can compare that anatomy to the anatomy of something that we see today. Okay. And hopefully we can look at a progression of the anatomy from that particular organism into what we see today, and see if we can look at a progression that shows us that evolution has occurred over a period of time to ultimately end up as the species that we're looking at today. So that's going to give us a progression that can hopefully give us a clue that evolution has occurred. And so what scientists can do when they look at fossils is they can take the age of a fossil using either carbon dating or radioactive dating to determine when that particular species existed. And so by taking a look at the age of that particular species, you can kind of date how far back that particular species existed and compare that to today's species. You take a lot of information, including the age, maybe other information in terms of the anatomy and things like that, and you compile all of that data into what's called the fossil record. And so scientists can use the fossil record to help identify what species may be related to what, or what species may have evolved into what current day species, and so on and so forth. Okay. So in order to do that, what scientists are going to be looking for is what's called a transition fossil. Okay. And so a transition fossil shows an intermediate anatomy between an earlier species and a current or a later species. Okay. So for example, here we have today's modern day horse on this side, okay? And then on the other side, okay, we have a species that evolved into that horse originally, which was called the Eohippus. Okay? And so the Eohippus was the kind of earliest form of the horse that we know of today. And so the Eohippus existed about 55 to 45 million years ago, and was kind of more like a dog-like ancestor. It was kind of about the size of a dog. It had paws like a dog. It looks kind of more like our classic dog. Now, in order for us to be able to know that today's modern day horse evolved from the Eohippus, one thing that scientists want to do is they want to look for transition fossils, or fossils that show kind of an intermediate anatomy between the Eohippus and today's modern day horse. Okay? And so what they ended up doing was they found some fossils of the Mesohippus, okay, which existed about 40 to 30 million years ago. And so the evidence that kind of indicates that the Eohippus likely evolved into the Mesohippus, which eventually became the horse, was that the landscape tended to change. Okay? So when we were looking at the Eohippus typically lived in a kind of a more forest-like environment, and so it only had shrubs and just things like different types of grass. And so it needed paws, and needed specific teeth shapes in order to be able to live in that environment. Okay. Then as the landscape changed, the Eohippus eventually evolved into the Mesohippus, okay, became a little bit larger. The teeth shape started to change in order to be able to eat the resources or eat the food that was available to them. And then the leg anatomy changed because, again, the landscape changed as well. Okay. And then so they started to develop some hooves. Okay. So that was about 40 to 30 million years ago, and again, they increased in size. And you can see that by looking at the fossils of the Mesohippus, which is classified as transition fossils between the Eohippus and today's horse. Okay. And then from there, the Mesohippus eventually evolved into the Hipparion, which existed about 23 to 2 million years ago. And so again, we just saw that the body size increased as the landscape again changed and the resources available differed. Okay. And so we saw increased body size, increased head size, different kind of jaw shapes. And then the Hipparion eventually became today's modern day horse. Okay. So again, in order to be able to know that the Eohippus eventually became today's modern day horse, we had to look for transition fossils, and specifically the fossils of the Mesohippus and the Hipparion, in order to kind of find that intermediate anatomy between the two to show us or indicate to us that the Eohippus eventually evolved into today's horse. Okay. So that's how fossils kind of indicate that evolution has occurred, okay, by looking for transition fossils between an earlier species and a later species. Okay. Now, another thing that we can do to kind of help to determine if evolution has taken place is to look at kind of the overall anatomy between existing organisms. Okay. And so the first type of structures that we can look at are called homologous structures. So homologous structures are structures that share the same basic form, their same basic kind of structure. Okay? However, they perform different functions. Okay? And so, for example, if we look at the kind of the human appendage, the dog appendage, the bird, and the whales, okay, the appendages are going to be built similarly. So here we're looking at the arm bones. Okay, so if we look at a human, we have the upper arm bone, the humerus, that's going to kind of help form two joints with kind of two bones here. Okay? And then we have the wrist, which make up our forearms. Then we have the wrist bones, and then the phalanges, which are our fingers. Okay? If we look at the dog is going to be shaped pretty much the same way. There's going to be an upper kind of leg bone here. There's going to be two bones. Then we have the wrist bones, quote unquote, and then we have the phalanges, which make up the paws. Okay? If we look at birds, similar thing. Okay, we got this upper bone that kind of forms the initial parts of the wings. Okay? Then we have the later parts of the wings. Then kind of some kind of wrist bones, and then the tip of the wing here. Okay? If we look at the whales, kind of same thing. Okay, one, and then we have a two, and then we have a kind of wrist bones, and then some phalanges here that make up the fins. Okay? So we can see just by looking at the bones, they're all built similarly, okay? Which indicates that there might have been a common ancestor between all of these organisms. Okay? And so the idea or the kind of theory is that there was some initial common ancestor. And so as the kind of the populations of that ancestor started to move to different areas, and they started to be exposed to different selection pressures. Okay? They might have been exposed to, some might have gone to kind of the water, some might have taken to the air, and so on and so forth. They went to different environments, and as they moved to different environments, they needed to be able to do different things in order to survive in that particular environment. Okay? They needed their appendages to be kind of more modified and more specialized. And so in the case of the human appendage allowed, for grasping and manipulating of different things in your environment, moving things from point A to point B. Okay? In the case of the dog's appendages allow the locomotion of that particular organism, of that particular species. Okay? In the case of birds, okay, these bones allow flight. And then in the case of whales, these bones allow swimming. Okay? So that even though they're all built similarly, they all have to be slightly modified, okay, in order to can allow that organism to best survive in the environment that it was in. So they developed the same structure, but then modified the structure a little bit in order to accomplish a specific function. Okay? So each one has a different function, but the same overall structure. Okay? Okay. So they have to be modified in order to fit the environment that they're in. Okay? So those are homologous structures. So same function or same structure, but different function. Okay, so another example of anatomy is to look at vestigial traits. Okay, so vestigial traits are traits that continue to persist from generation to generation, even though they do not have a function. Okay, so for example, here we've got an ostrich. Okay, an ostrich is a part of the bird family, and most birds are going to have wings that allow them to be able to fly. Okay, so most birds have wings for flight. Okay, in the case of the ostrich also has wings. But the wings are non-functional. Okay? They don't allow the ostrich to fly. Okay? And so the wings on an ostrich, because they don't have a function, are considered vestigial traits. Okay? They're traits that continue to exist and continue to persist from generation to generation, even though they do not have any function. Okay? So the wings on flightless birds are examples of vestigial traits. Another example would be, if we're looking at the pelvic bones on snakes. Okay? So snakes are part of the reptile family. And so most reptiles will have hind legs that are going to have pelvic bones attached to them. Okay. So if we look at frogs, or we look at other lizards and things like that, they're going to have a pelvic bone with some back legs attached to them. Okay. Snakes also have pelvic bones, okay, but obviously we know that they don't have any legs. Okay. So that usually will indicate to us that there must have been a reptile ancestor that had a pelvic bone and had legs. But then as snakes started to kind of evolve from that, they lost their legs, but maintained their pelvic bones, okay? Even though their pelvic bones do not serve any function, because again, they don't have any legs. Okay? That's another example of vestigial traits there. Okay? Some cave animals, and typically cave animals that live in, that live deep in the sea, will have some eyes, so they're also gonna be vestigial traits because those eyes are not going to be functional. Okay? They're still gonna have two eyes, but the eyes either don't have either are not able to work, just because of their own anatomy, or they're just not able to work because they're so they live in such dark environments. Okay? And so those eyes would be non-functional, so those eyes would be considered vestigial traits as well. Okay? They can continue to exist even though they do not have any function. Okay? Those animals are effectively blind, okay, despite having eyes. Okay? Cool. So those are vestigial traits. Okay. Any questions on vestigial traits or homologous traits? Okay. Okay. So then the last one is called analogous structures. Okay. And so analogous structures are structures that perform the same function, okay, but are built differently. They have a different structure. Okay. And so this is evidence of what's called convergent evolution. Okay. So convergent evolution basically tells us that organisms will or kind of start to develop or evolve similar, or how do I say this? They evolve traits that perform the same function because they exist in the same environment. Okay? So an example of this would be kind of the Arctic fox and the ptarmigan bird. Okay? They both are going to have white coverings. Okay? So in the case of the Arctic fox, it's going to have white fur. In the case of the ptarmigan bird, it's going to have white feathers. Okay? They're built differently. They have a different structure. The fur and the feathers have different structure, okay? But they perform the same function in terms of helping these organisms to blend in with their environment and not be easily seen by predators. Okay? So even though they're built differently, they evolve structures that perform the same function. Okay? And the reason for that is because they share the same environment. Okay? So because they both are going to need to blend in with the snows in order to escape predators, they're both going to need to develop white coverings, but their coverings are going to be different. Okay? Performing the same function, but they're going to be different. So that's what we call convergent evolution. So convergent evolution is where you develop traits that perform the same function, okay? Because you share the same environment. So that's going to be different from what we saw with homologous structures, which are going to be evolution or evidence of divergent evolution. Okay? So that divergent evolution tells you you have a common ancestor, and the common ancestor ends up diverging into multiple different other species in order to kind of to show like an evolutionary relation or using a common ancestor. In the case of convergent evolution, there is no common ancestor, okay, but they do develop traits that perform the same function just because they exist in the same environment. They're exposed to the same selection pressures, so they need to have kind of traits that perform the same function to one another. Okay. Does this make sense? So it's kind of all over the place on this one. Does analogous structures make sense? Okay. Okay, so analogous structures, you have different structures that perform the same overall function. Okay. Okay. Another thing that we can do to kind of determine kind of relatedness between different species is by looking at the embryology. Okay. So embryology is a study of how an organism develops from the time that they're a zygote to the time they become an adult. Okay. Now, the one thing that we're really going to be focusing on with embryology is how these organisms will develop before they're actually born. Okay. So if we kind of look at the fish, if we look at the salamander, the tortoise, the chick, the rabbit, and the human, okay, they're actually all going to kind of develop similar structures kind of before they're born. Okay. So all these organisms are going to have a gill slit and a tail, and then a few other structures as well. Okay. Sometimes a forelimb bud and a hind limb bud, okay, which will eventually become the arms and legs. Okay. And so what's interesting is that humans actually have gills when we're first developing in the uterus for the first few weeks of our time in our mom's womb. Okay. And so many organisms will share these same characteristics, having a tail, having gill slits, and then as they continue to develop, sometimes they'll lose different aspects of it. Okay, so in humans, we end up losing our gills, and then we lose our tails, and then eventually we become kind of the overall structure that we see on each other on a day-to-day basis. Okay, same thing with the rabbits. Okay, rabbits will ultimately lose their gills as well, and then in some cases, their tails are going to be much more kind of smaller. Okay. Same thing kind of with the chick, the tortoise. The salamander and the fish will maintain their gills and their tails, but they'll kind of have a reduced forelimbs and hind limbs as well. So there's going to be certain structures that are going to be present as we're embryos, okay? And then they become absent as we start to develop later into our childhood and adulthood. So this can typically be an indication that some of these organisms may have some relatedness to one another as well. And then as scientists, you can kind of go in and try to figure out how much relatedness is actually between all of these different organisms. Okay. That's embryology. Okay. And then another technique that we can use to use as evidence for evolution is to look at the molecular biology, and more specifically, look at the DNA. Okay. So every organism is going to have its DNA, okay? And the DNA is going to work exactly the same across the board for every species. And at the same time, the genetic code that we saw earlier to help us produce various types of proteins and things like that, to help us activate specific genes, that's also going to work exactly the same across the board from species to species. Okay? So DNA replication and gene expression are all going to work exactly the same. Okay? And so what we can do using that information is we can actually look at the DNA sequences between different species and look at the similarity, or how much similarities they have, how much similarities different species will share, to kind of show that they're related to one another. Okay? So the more similarities that are in the DNA, okay, the more likely those two species are going to be related to one another. Okay? So for example, if we look at human DNA and then chimpanzee DNA, okay, our DNA is about 96% similar. Okay? And so we know that humans and chimpanzees are related to one another, okay, even though we may not have, we're obviously not the same. Okay? And so that 4% difference is really all that's necessary to kind of create the very vast differences between humans and apes. So again, that the more similar or more related species will share more of their DNA, okay, which makes sense. And then more distantly related species are going to share less of their DNA with one another. Okay? So for example, if we were to kind of compare the DNA between kind of human and a tree, okay, our DNA sequences are going to be very different because we're not related to one another. Okay, so you can look at the DNA sequences to kind of help you identify, how closely related some species may be to one another. Okay. And so not only can you have evolution of the species themselves, but you can also have evolution of certain genes that will also ultimately lead to specific proteins becoming kind of evolved for a specific function or structure as well. Okay. And again, all that's going to be taking place specifically inside the DNA. So, question here. So birds, bats, and bees all have wings that allow them to fly. Okay? So looking at the wings, we can see that they all look different. Okay? How would we classify the wings in these organisms? We have A, they would all be considered homologous structures. B, the wings would be considered analogous structures. C, they would be considered vestigial structures. D, all the above, or E, none of the above. How would we classify these wings here? Okay, so Jackie says analogous. Anybody else? Anyone agree? Anyone disagree? Yeah, so Christopher says B as well. Yeah, and B is correct. These would be analogous structures. Okay. So analogous structures, again, they have different overall structures, but perform the same function. Okay. So in the case of bats, birds, and bees, okay, all of these wings are going to allow them to be able to fly, okay, but they're all going to be built differently. Okay. Those are going to be analogous structures. Okay. Different structure, but the same function. So this is evidence of convergent evolution. Cool. All right, any questions on anything we've talked about so far? Okay. All right, so if not, that's going to lead us to our next chapter, or our next section here, which is speciation. Okay. So we're going to be looking at how different species can actually come about. And how do we actually produce new species from, kind of one pre-existing one? Okay. And so when we look at a species is defined as a group of interbreeding individuals. Okay. So if you have two individuals that are able to breed with one another and produce offspring, okay, then we know that they are of the same species, that they're one species. Okay. However, if we have two individuals that cannot mate with one another, or that they can mate and not produce any fertile offspring, okay, then we know that they are a different species, okay, although there are some exceptions to that. Okay. So an example would be kind of the African elephant and the Asian elephant. Okay. So originally, before kind of these two kind of evolved into their own individual species, there was one population of elephant kind of around kind of Asia and Africa. Okay? There was one population of elephants. Okay? Now, as those elephants started to move to different areas, some started to move deeper into Asia, other ones started to move deeper into Africa. They were exposed to different selection pressures. Okay? They were exposed to different environments and different resources, and they ultimately started to develop into different traits. And so they were eventually separated for such a long period of time that they eventually became kind of different, slightly different species here. See the African elephant that has more wrinkled skin than the Asian elephant does, and the Asian elephant has much smaller ears than the African elephant does. Okay. So they eventually evolved into new species, and so if you try to bring them back together with one another, if you try to take an African elephant with an Asian elephant, and you try to have them mate with one another, they won't mate with each other. Okay. And so because they won't mate with each other, that's evidence that they're now two different species. Okay. They are not the same anymore. They're two genetically different, and two kind of obviously now phenotypically different, and so they won't mate with each other. Okay? So they're two individual species. Okay? So this is the process of what's called speciation. Okay? So speciation is the process of forming two species from one original main species. Okay? Now again, there are some exceptions to this rule. So if you think about kind of forming a liger, where we have a lion mating with a tiger, and we form that liger, okay? Obviously those are two individual species, okay, that produce an offspring. However, the offspring that it's able to produce, that liger, is no longer fertile, so it cannot produce any offspring of its own. Okay? Same thing with if you try to mate a donkey with a horse and you get a mule. Okay? Again, two different species, they mate with one another, but the mule itself cannot create its own offspring. Okay? So again, those are a couple of exceptions to the rule, but that's kind of the general rule of thumb. Okay. That's how speciation will typically work. Okay. Now, for speciation to take place, we are obviously going to be forming two new populations from one original population. Okay. And for us to be able to know that there are two new species, it must be impossible for those new populations to interbreed with one another. Okay. They cannot form any offspring with each other kind of naturally. And so there's two different mechanisms for speciation. We have what's called allopatric speciation, and we have sympatric speciation. Okay. So these are the two ways that we can end up with different types of populations in different species. So the first one we're going to look at is what's called allopatric speciation. Okay. So allopatric means other homelands. Okay. Allo meaning other, and then patric meaning homeland and things like that. Okay. So allopatric speciation is where you have an original population that ends up getting divided by some geographical barrier. Something physically separates those two populations from one another. Okay? And so when they get separated, okay, they're no longer able to cross that barrier, and so they just breed with one another, and they have no way of kind of crossing across that barrier. Okay? So because they get exposed to different selection pressures and different things in their environment, they eventually will if they're separated for long enough, they eventually will kind of diverge into different directions, and then ultimately result in two different populations, okay? Or two different species. Okay? So here's an example. So here we have a population of fish, and let's say the water level drops, and then we have this piece of land that separates these two fish from one another. Okay? Well, the fish on the left side might be exposed to different selection pressures. They might be exposed to like different temperature and things like that, different food sources, okay? Than the fish on the right side. So again, if they're exposed to different environments and different selection pressures for long enough periods of time, they will eventually become their own individual species. They'll only pick up on different traits that allow them to be able to survive in one environment. The other one is going to have other specific traits that allow them to survive in their own environment. And again, they're going to kind of diverge in opposite directions and become new species. Okay. So that is what's considered allopatric speciation, where they get separated by a geographical barrier. So that's what happened here with the porkfish. Okay. So back about more than 3.5 billion years ago, there's a population of porkfish that existed around here in the Caribbean Sea. Okay. And so the porkfish were just kind of floating throughout their population. They're just kind of swimming back and forth. And so we just had just one population of porkfish. Okay. However, about 3.5 million years ago, the Isthmus of Panama rose out of the ocean and actually physically separated these two the porkfish population into two areas. Okay. We separated some of the porkfish on one side, on the eastern side. The other ones were kind of separated onto the western side. Okay? And so because they were separated by that geographical barrier, each one on each side, or each set of porkfish, became exposed to different environments. Okay? Maybe there was a colder temperature or a different food source and things like that. Okay? They were exposed to different selection pressures, and so they needed specific adaptations in order to survive in that particular environment. Okay? So as they developed those different adaptations, they eventually developed into their own individual species. Okay, where we have just the original porkfish population, and then we also now have the Panamic porkfish population. Okay? And so this was all due to the separation by this Isthmus of Panama that actually physically separated the initial population of porkfish. Okay, so that's what we consider allopatric speciation. Okay, separation by a physical geographical barrier. Okay. So we'll kind of come back to that one, and that'll hopefully make more sense as we kind of go through this a little bit more. Okay. The second type of speciation is what's called sympatric speciation. Okay. So sympatric speciation is where you have an original population here, okay, and there's some individuals in that original population that start to develop some mutations. Okay. And so as they produce those mutations, they eventually will become their own individual species, okay, and they're only able to mate with their own species. Okay. And so we end up having a few others starting to develop until you ultimately have kind of one new species developing in the same area as the original population. Okay. So this is where you have speciation taking place in the same kind of geographical area, in the same location. So this is usually going to be due to changes in or kind of mutations or changes in chromosome structure. We'll look at this in a little bit more detail as we go forward too. So this would be kind of an example here. So here we've got some trees. There's some trees here. There was some kind of genetic mutation that happened that ended up resulting in, trees that look like this, that are a little bit lighter. Okay? And so because the genetic structure of those new trees is different from the original ones, they're no longer able to mate with one another. Okay? They have to mate with their own individual species. And so you end up with new species developing, okay, from an original population in the same area that the original population was already in. Okay? So sympatric speciation is where you have speciation in the same geographical area. Okay. So again, this will hopefully make more sense in a little bit too. So an example of what happened with sympatric speciation is when we were looking at some fruit flies. Okay. So about 200 years ago, in about the 1800s, there were some fruit flies. Okay. And across North America, there was a lot of these hawthorn trees. Okay. As the hawthorns kind of look similar to apples, but they're not apples. Okay. They're hawthorn. Okay. And so these fruit flies will start to kind of mate and kind of live on these hawthorn fruits. Okay, because that was the environment that they were on. They had their own kind of traits, their own adaptations that allowed them to be able to survive on hawthorn trees. Okay. However, around the 1800s, or the late 1800s, there were some kind of European settlers, and they started to bring apples in from Europe. Okay. And so they started to plant apples in the same area as they did with the hawthorns. Okay. So we had hawthorn trees and we had apple trees now kind of occupying the same general area. Well, the fruit flies that were initially on the hawthorn trees occasionally started to develop some mutations in their smell, okay, in their smell receptors. Okay. So instead of kind of spending most of their time on the hawthorns, they started to move and be more attracted to the apples and the apple trees. Okay. And so again, the apple trees and the hawthorn trees in the same general area. Okay. So we started to have some kind of hawthorn flies and then some apple flies. And so because the apples kind of produced or kind of created a new environment and new selection pressures for the fruit flies, okay, these fruit flies needed to develop their own adaptations in order to survive better on these apple trees. Okay. And so what ended up happening is we saw evolution of the fruit flies. There are some fruit flies that have specific adaptations for hawthorns, and other fruit flies that have specific adaptations for apple trees, depending on which trees they spent the majority of their time on. Okay. And so because they spent their time on different trees, they were exposed to different selection pressures, and because they never really went back to the opposite tree, they stayed where they were at, they eventually became their own individual species. Okay. And so the hawthorn flies and the apple flies were completely different species at this point in our current day. and they will not mate with one another because they're now so genetically different because of the environments that they had spent the majority of their time on. Okay. So that's an example of sympatric speciation, where you had speciation happening in the same general area, same general location. Okay. Does this make sense? And how sympatric speciation works here? Everyone kind of following me here? Yeah, okay. Cool. Okay, so what we're going to do first, we're going to kind of dive a little bit deeper into allopatric speciation, or where we have speciation through geographic separation. Okay, and so typically what initially happens is we have a continuous population, a geographically continuous population, and so you have free gene flow across an entire population. Okay, you have a relatively homogeneous gene pool. There's a good mix of all the genes located throughout that particular population. Okay, so you have free gene flow, free mating, everything is hunky-dory. You have allele frequencies that are evenly spread throughout that population. Okay, that's in the original population. Now, when that population gets separated by geography, whether it's the water levels dropping and getting a physical barrier, or if you have a new valley showing up, or if you have for example, kind of a new river that gets developed that physically separates that population into two individual populations, okay, you interrupt the gene flow. Okay? The gene flow gets interrupted. You no longer have individuals that are able to cross over that barrier. Okay? And so you end up forming two individual populations. Okay? Those populations are then going to be exposed to different selection pressures. And as they get exposed to different selection pressures, okay, their adaptations are going to start to develop for the environments that they're in. Okay? And they're going to evolve in different trajectories, based on the habitats that they are now divided into. Okay. So by doing that, the allele frequencies end up changing, and that ultimately will lead to the evolution of those particular species. Okay. So an example of this was with the antelope squirrel around the Grand Canyon. Okay. So originally there was one kind of general antelope squirrel population. And so the antelope squirrels, they kind of just existed. They were kind of homogeneous. They had free gene flow. Everything was good. They were able to mate freely, and everything was kind of hunky-dory. Everything was cool. Okay. Now, as the Grand Canyon started to separate, and as we started to kind of develop the Grand Canyon, those populations of antelope squirrels ended up getting separated. Okay. And so as they got separated, they were exposed to different selection pressures. And so those selection pressures caused them to start to develop different adaptations, and ultimately caused them to evolve in different trajectories. Okay. And so we ended up with the Harris antelope squirrel, which has this kind of white stripe along its side and more of a longer, bushier tail, okay, and a larger body. Okay. Whereas the white-tailed antelope squirrel, okay, has a wider tail. Okay. It has a smaller body. It still has this kind of white stripe. It's a little thinner, and it's a lighter color. Okay. So because of these selection pressures that they were then kind of separated into, or divided into, they developed their own adaptations in order to help them survive in that particular environment, and ultimately led to their evolution. Okay. So again, if you try to take the Harris antelope squirrel and the white-tailed antelope squirrel and you try to bring them together, they won't mate with each other. Okay. They're completely different species at that point. Okay. Okay. So like I said earlier, if those populations get separated for a long enough period of time, each one is going to get exposed to different selection pressures, and those selection pressures will cause the evolution of that particular species. Okay. So each population, as they're exposed to their own selection pressures, will evolve into different trajectories and then become their own species. Okay. So question for you guys, kind of a more kind of open-ended question. What are some kinds of selection pressures that might result in evolution? And what do we think are some things that we might find in the environment that can cause evolution to take place, different selection pressures? Okay. What do we think? So Kimberly says food. That's a good one. Let me go ahead and kind of write these ones down. Okay. So we got the food. Okay. Okay. Food is going to be one. Okay. Weather, right? Weather is going to be another one. Okay. What else? The habitats. Exactly. The habitats. Cool. Okay. What else? What are some other selection pressures? Predators. Yeah, that's a good one. That's a big one. So predators. Anything else? Mating choices, yes, absolutely. So mating choices are going to be a big one. Okay. What else? Actually, I think what I'll even do is I'm going to take mating choices a little bit further, and I'm going to go ahead and say mating behaviors as well. Okay. Cool. Can we think of any others? Water sources, yeah. Cool. Okay. Anything else? Yeah, so these are kind of the major selection pressures, right? So all of these are the major selection pressures. So the food is going to be the food availability and what types of food are available to them to eat. And those are going to be some selection pressures. If you're able to eat one kind of food and not another, and that's going to help to create or help to encourage different adaptations to develop, and then ultimately lead to evolution. The weather and the kind of climate that you're in, okay, that's going to lead to different evolution possibly as well. Okay. The habitat that you're in, any predators that may be available in that specific environment. And you may need specific traits that allow you to be able to escape from those predators. Mating choices and mating behaviors. You might some organisms will develop specific dances or specific kind of mating rituals in order to help them kind of attract mates. Okay. Access to water. Okay, that's going to be another one. So all of these different selection pressures can ultimately lead to different organisms developing different adaptations to allow them to access those resources, okay, and be able to survive and ultimately reproduce and pass on those adaptations to their offspring. Okay? And as they do that, that'll ultimately end up leading to evolution. Cool. All right, good. So those are all kinds of different selection pressures. Cool. So that's good. So as this takes place, as we start to develop some of these adaptations, in order that are kind of result from this geographic separation, the allele frequencies end up shifting. They end up changing. In some cases, you might end up going from like 50% of one allele frequency, and then after that separation, now you have like 80% of that particular allele that's present, okay? And then 20% of the opposite allele. Okay? So the allele frequencies are going to shift. We can see some mutations begin to develop, which can also alter the allele frequencies as well. And then the other thing that can take place is genetic drift can also play a larger role, because now you're dealing with a smaller population. And because you've decreased the population size and you've separated your populations into two, okay, you're dealing with smaller populations, and so genetic drift can play a larger role in shifting those allele frequencies, okay? And then ultimately leading to the development or adaptations or development of different traits and adaptations. Okay. So as that separation continues to develop, and as we continue to keep those different populations separated from one another, genotypic changes are going to occur over a longer period of time that are going to ultimately lead to phenotypic changes. Okay? And if those changes, ultimately lead to kind of changes in the reproduction, okay? If they lead to changes that influence reproduction in any way, that can lead to what's called reproductive isolation, okay, which means that there's going to be an inability of those individual populations to breed with one another. Okay? Yep. So that can be due to just kind of different genes changing within the populations, or and again leading to different phenotypes, or it can lead to genotypic changes that cause changes in the reproductive either behaviors or reproductive actions that will ultimately also lead to reproductive isolation. Okay? And so when we have reproductive isolation, and we'll talk about what that means in a second, okay, when we have reproductive isolation, that will ultimately cause mating to be less likely, because again, you're going to be physically separating those two populations from one another. Okay, but in addition to that, they may become so different from one another that they're not going to recognize each other as the same species. Okay? And so they just don't mate with each other, because it's almost like they envision it as trying to mate a human with a dog or something like that. It just doesn't work. So they're not going to even bother. Okay? And so that can happen. The other thing that can happen is with offspring, the offspring that you may produce, if you try to mate two different species with one another, okay? If you are able to produce offspring, the offspring can either be non-viable, meaning that they don't survive to birth, or they're infertile themselves. Okay, so they're able to be born, but they themselves cannot produce their own offspring. Okay, so those are the two things that can take place. So we've got two types of reproductive isolation. Okay? The first type is called pre-zygotic reproductive isolation, which just means before fertilization even happens. Okay? When we talk about before fertilization, we're even talking about before they even attempt to mate with one another. Okay? So there's different things that can lead to pre-zygotic reproductive isolation. The first one is like timing of the mating. So for example, there's certain species, I think like the American bullfrog or the American green frog and the bullfrog, they mate at different times of the year. Okay, so some might mate in the earliest summer, some might mate only in the late summer. Okay? So because they're mating in different times of the year, they're never going to come into contact with one another, and they're never going to mate with each other. They're never going to attempt to mate with one another. So the timing of the mating, either in different seasons or throughout the entire year as a whole, okay, can prevent them from mating with one another. The number two is that they may be being exposed to different pheromones. Okay? So for example, there's some pheromones are just different types of hormones that can get released, and different molecules that get released that indicate that you're ready to mate. Okay? And so some species may release different types of pheromones that another one is going to be able to recognize. And so because there's a difference in the pheromone production and pheromone kind of recognizing, they're not going to be able to mate with one another because they're not going to recognize each other's pheromones. Okay? The third place might be mating sites. Okay, so for example, you might have some birds that want to mate higher up in trees, and then other birds that want to mate lower in trees. Okay, so because they're going to be mating in different areas, okay, different mating sites, okay, they're never going to come into contact with one another, and they're never going to mate with each other. So that's another one. Different mating rituals. So again, some organisms will have some specific dances that they'll do to kind of court the females. And so if you have one species of bird and being exposed to another species of bird, this bird might be kind of doing its own mating dance, okay? But that mating dance is going to look completely weird to the other one, and it's going to say, okay, that looks weird, I don't want to mate with you, you're weird, I'm cool, I'm gone. Okay? And so because their mating rituals don't line up, okay, they don't mate with one another. Okay? So again, kind of helps them form different species. Okay? And then again, female preferences might be different, male behaviors might be different, which can cause, again, differences in the reproductive isolation there. Okay? So those are all pre-zygotic reproductive isolation, things that happen before they actually have a chance to mate with one another. Okay? The flip side of pre-zygotic reproductive isolation is what's called post-zygotic reproductive isolation. So this is where you have two different species that are actually able to mate with one another, and then we'll see what happens after that actual mating process happens. Okay? So this is after fertilization has taken place, after you form a zygote. Okay? And so the first that can happen is we can have some genetic incompatibilities between the parents. Okay? Basically what that means is that the genes between the parents are not compatible with one another, and so they're not able to produce an offspring. Okay? So they may be able to mate, but they're not going to be able to produce an offspring. Okay? That's number one. Number two is that the offspring that do get produced may be non-viable. Okay? So you may be able to mate with one another and then produce an offspring, okay? But that offspring is not able to survive to birth. Okay? And so it ends up dying before it has a chance to be born. Okay? So again, you end up forming that new species because, again, that species never got a chance to survive or reproduce, okay? Or that new one never got a chance to survive and reproduce. Okay? Then the third one is you may be able to produce an offspring, okay, but the offspring itself may be sterile or infertile. Okay? And so what that means is that the sperm or the eggs, the gametes themselves are non-viable, and so they're not able to produce their own offspring. Okay? So an example would be like the donkey and the horse. Okay, these two are different species, but they are able to mate with one another. Okay? And when they mate, they form the mule. Okay? The mule, okay, is able to survive, but that mule is not able to produce any viable gametes, not able to produce a sperm or not be able to produce eggs. So in doing so, it's not going to be able to reproduce. Okay? So that mule basically stops at itself. Okay? It's not going to be able to produce any more offspring from there. Okay. So those are the reproductive isolations. Okay? Any questions on reproductive isolation? Okay. Okay, so this geographic separation, so this isolation leading to geographic separation can form in a number of ways. We may see a river that kind of forms a new branch somewhere, and when that river forms, it divides two individual populations from one another. You can have erosion that forms a new valley. And as you form that new valley, again, you now separate two populations from one another and they get exposed to their own selection pressures. Or it can be due to organisms traveling to a new area and they don't have the ability to get back to where they originally were. Okay? So again, that can lead to isolation as well. Okay. So that being said, the ability of geographic isolation to kind of isolate those two populations is going to depend on the organisms themselves as well. Okay? So even though you do have some isolation taking place, the kind of the ability for that to lead to speciation depends on the organism and the organism's ability to move. Okay? If it's able to get back to where it initially was, then most likely we're not going to see any speciation occur because you're going to be able to have the easy gene flow from one spot to the another. Okay? However, if you're not able to get back, okay, then that's going to lead to some speciation occurring. Okay? So an example, or the two different types of geographic separation are called dispersal and then vicariance. Okay? So in dispersal, what we have is individuals from one population, okay, moving, and they kind of move to another area, okay, which you see over here. Okay, they move from this mainland over here to this island, for example. And as they move to that island and they don't have the ability to get back, or they just don't go back, okay, they end up forming their new populations and new species because they're being exposed to different selection pressures, okay? Either different temperatures or different climates, different food sources, different predators. Okay? They get exposed to different selection pressures and they end up forming new adaptations and ultimately become new species. Okay. The other thing that can happen is what's called vicariance. And so vicariance is where there's a physical barrier, okay, that arises that separates the initial population. Okay. So here we have species A. Okay. It gets separated by this river, for example, this barrier here. Okay. And so these species have no ability to get across this particular river. Okay. And so as they get exposed to their own selection pressures, okay, they eventually become two different species, okay, species B and species C. Okay. So there's a barrier that separates them, and as they get separated, they get exposed to different selection pressures, and they ultimately become their own individual species. Okay. So it's dispersal and vicariance. Okay. So question here. So based off of what we just learned, the Isthmus of Panama arose after the appearance of the porkfish. Okay. So is this an example of dispersal or vicariance? What do we think? So Josie says B. Tim says B as well. Yeti says B. Yes, that's absolutely correct. B is the answer here. Right, so B, this would be an example of vicariance, again, where we have this kind of barrier here. And as the barrier separates our initial population, we're ultimately going to end up with two new populations, okay? Two new species. Cool. Okay, so now, based off of what we learned here, okay, which of the following is most likely true about speciation and distance? Okay, so A, the further apart populations are, the more likely speciation will take place. B, the further apart populations are, the less likely speciation will take place. Or C, the likelihood of speciation taking place is independent of distance. Okay, what do we think here? Okay, based off of what we've learned so far. Which one is most likely true? So Yetzi and Tim both say A, and A is the correct answer. Right. So perfect. Good. So the further apart populations are, the more likely speciation will take place. Okay. Now that being said, why do we think this is the case? Okay. Why would this make sense? Why would we get more likely speciation the further apart our populations are? What do we think? Anybody? So Kimberly says the changes in the environment. Yes. So they don't, Christopher says they don't mate with each other. Okay. Yetzi says there are less similarities. And you guys are all absolutely correct. Okay. Those are all going to be major, contributors to creating different species. Okay. And so basically what we see here is as the distance begins to increase, there's going to be less similarities in the environments. And there's going to be less, there's going to be a lot more changes in the environments. And they're not going to be able to mate with one another. Okay. So for example, the ecosystems are going to be different. They're going to be exposed to different types of prey. They're going to be exposed to different types of predators. They're going to be exposed to different types of climates. They're going to be exposed to different kinds of water availabilities, things like that. All the selection pressures are going to be most likely to be very much more different the further apart you go. Okay. And so again, here you end up with much different selection pressures. If you have different selection pressures, that will ultimately lead to the evolution of new species. Okay. So for example, I forget how long ago, I believe it's probably about 10,000 years ago as well, there was a population of owls. Okay. So there was an ice age that happened, and there was a large glacier that separated the owls on the western side of the United States as compared to the owls that are in the southern United States as well as into Mexico. Okay. So what we ended up with is the northern spotted owl and the Mexican spotted owl, because they both were exposed to different environments. Okay. The northern spotted owl was exposed to more kind of colder environments, different prey, different predators and things like that, while the Mexican spotted owl was exposed to more warmer climates, okay? And again, different prey, different predators, different ecosystems and things like that. So because they're so far apart, they eventually were exposed to different selection pressures. Their selection pressures were a lot more different from one another, and that ultimately led to the production of new adaptations, which ultimately led to evolution of that particular species, of the spotted owl. Okay. Everyone following me so far? This makes sense. Yeah, okay, cool, good. Okay, so there is another kind of specific type of geographic separation here, or allopathic speciation, which is called adaptive radiation. Okay, so adaptive radiation is where you have a member of members of one population kind of in one particular area, okay, and then those members start to disperse to multiple different other places. Okay, so they move to different other places. Okay, multiple different areas all at the same time, okay? And then they form new populations in those particular areas. Okay? Now, as they get exposed to their new environments, they're going to be exposed to new selection pressures. And as they get exposed to new selection pressures, they'll eventually develop new adaptations and then eventually become their own species. Okay? So this is what we saw with the Hawaiian honeycreeper species. Okay? So there was initially one kind of Hawaiian honeycreeper, and as this particular bird started to move to different islands of Hawaii, each bird, or each kind of the different bird populations were exposed to different environments, different selection pressures, different environments, different climates, different prey, predators, so on and so forth. Okay? And so because they got exposed to different selection pressures, they needed to take on specific traits. They needed new adaptations in order to survive in that particular environment. And so ultimately that led to the development of these adaptations in different islands of Hawaii. Okay? So the bird at the Liwi, we had the Apanani, the Nihoa finch, the Maui, Maui parrotbill, and so on and so forth. So they all developed their own individual traits, but they initially all evolved from this one Hawaiian honeycreeper that was initially on the main island. Okay? So basically you have one founder species, okay, that evolves into many species all at once, okay, as they start to move to different environments and different populations. Okay, so that's what we call adaptive radiation. Okay, so that's allopathic speciation, okay, or geographic separation. Okay. Any questions on allopathic speciation or speciation using geographic separation here? Okay, so that's going to lead us to speciation that happens without geographic separation. Okay, so this is going to be where we have sympatric speciation occurring. Okay, so in sympatric speciation, this is a process that's a little bit more complicated than allopatric speciation, and this is typically going to be due to changes in the chromosomes. Okay? Specifically, what's going to be happening with polyploidy. Okay? So if you remember going back to chapter 8, when we were first looking at polyploidy, polyploidy typically happens when we have errors in our chromosome number. Or you have an extra set of chromosomes. Now, this is typically going to be lethal in animals. Okay? Animals and humans cannot really survive polyploidy, typically. Okay? However, polyploidy actually works pretty well in plants. Okay? And so we have different types of polyploidy. The first type is what's called autopolyploidy. Okay? So autopolyploidy is where you have extra chromosomes from your own individual species. Okay? And so the way this happens And you don't need to really get into the nitty gritty here, but basically what happens is we have some diploid plants, and instead of producing haploid gametes like we saw during meiosis, they end up producing diploid gametes instead. Okay? And so the diploid gametes that end up getting produced, okay, are no longer compatible with the haploid gametes that are normally produced. So they cannot mate with one another. Okay? Diploid gametes cannot mate with haploid gametes. Okay? So instead what happens is the diploid gametes are kind of forced to self-fertilize with each other. Okay? They self-fertilize with themselves and produce their own individual species, okay? And mate with themselves and produce this new species that we see here. Okay? That's autopolyploidy. Okay? You end up producing your own chromosomes from your own individual species. Okay? Diploid gametes cannot mate with haploid gametes. They can only mate with other diploid gametes. And so you end up forming a new species because you have different genetics. Okay? Okay. That's that. Okay, that's autopolyploidy. Okay? The other type of polyploidy that we can have is called allopolyploidy. And so this is where you have individuals from two different species that mate to produce some viable offspring. Okay? So here you have one species, okay, that has its own set of chromosomes. You have another species that has its own set of chromosomes. Okay? They produce their gametes. Okay? The gametes look different from one another, okay? And then they mate. Okay? Then from there, these gametes then self-fertilize, okay? And as they self-fertilize, they end up producing a normal gamete and then a polyploid gamete, okay? Which will then again mate with one another and then produce a new offspring. Okay? So this new offspring now becomes a second species or a new species here. So this requires 2 generations to occur. Okay, don't worry too much about this for the exam. This is a more complicated version of for to see how sympatric speciation occurs. But just know this can happen as well. Okay? So this is something that's typically seen in wheat plants and cotton plants and tobacco plants as well. Okay. And so that's usually due to some kind of error in meiosis. Now, another way that we can have sympatric speciation is if we have kind of individuals occupying different aspects of the environment. Okay? So for example, here let's say we've got our body of water and we've got some fish. Okay? These fish are all of the initially of the same species. However, we have some fish that spend some of their time in one part of the water and other ones that spend in another time, or their time in another part of the water. Okay? So let's say we have a subpopulation of these fish that spend most of their time here at the surface. Okay? And then we have another kind of subset of the population that spend their most of their time here kind of more deeper in the water. Okay? So because each of these fish is then going to be exposed to different selection pressures, so let's say the top fish are going to be exposed to different types of food, the temperature is going to be different, the predators available are going to be different, the prey available to them is going to be different. Okay? They're going to be exposed to different selection pressures than the ones that are below. Okay? The ones that are below, that are deeper, might be exposed to different amounts of light. They're going to be exposed to different predators. They're going to be exposed to different food sources. Okay? Their selection pressures are going to be different. Okay? So even though they're occupying the same general location, okay, the same general area, okay, they're going to end up developing into different species because of the selection pressures that are found in the different locations that they're in. Okay? They're in the same body of water, but some are going to occupy the surface, some are going to be a little bit deeper, and they're going to have different selection pressures, even in that one kind of geographical location. Okay, and so as they're exposed to different selection pressures, they're going to eventually develop different adaptations, and then eventually to the evolution and the speciation that takes place here, okay? Just by being exposed to different parts of that same body of water. Okay. So that's where we can have sympatric speciation occurring as well. Okay, so another example of this happened with the cichlids in Nicaragua. So there's some cichlid fish here, and so even though they're exposed, they live in the same general area in Nicaragua. They eat different food sources. Okay? And so some cichlids will eat one type of food source, which will cause them to be more thin-lipped. There's other ones that are going to eat different types of food. And so as a result, because they're exposed to that one type of food source, they have to develop more thick lips, okay, in order to be able to consume their food. Okay. So they eventually become their own individual species. We have the thin-lipped cichlids as well as the thick-lipped cichlids. Okay, that's how speciation can occur. Okay, any questions on speciation before we go forward here? Okay, so the last topic that we're going to be looking at are the misconceptions about evolution. So we'll wrap up with this. And so when we talk about evolution, there's a lot of misconceptions that happen kind of on a day-to-day basis when people start to bring up evolution. So this last section is going to be used to kind of clear up some of those misconceptions about evolution, so you kind of think about it in a more kind of accurate light and have those discussions in a more appropriate manner. Okay. So the first misconception is that individuals evolve. Okay? Individuals in a population cannot evolve. Okay? Individuals do not evolve. Populations do evolve. Okay? So if we're looking at individuals, okay, individuals obviously are going to be changing as they go through life. Okay? But all individuals are going to be born with specific genes, and those genes will ultimately lead to them developing specific phenotypes and different physical characteristics. Okay? Those individuals are not going to be adapting. Okay? The development of those characteristics is not going to be evolution. Okay? That's just normal development. Okay? Individuals are not going to evolve or adapt to a given environment. Okay? So for example, let's say you go outside and you start tanning. Okay? That tanning, okay, you're getting a darker skin, is not evolution. That's just something that your genes are programmed to do. Okay? So that's just a normal development, a normal process that your body is able to perform. Okay? So when we look at adaptations and evolutions are the result of natural selection happening for multiple generations. Okay? And so humans, not humans, individuals can develop, okay? They can grow, they can undergo reproductive changes and things like that, but they're not going to evolve. Okay? Evolution and adaptations are going to develop over multiple generations as we start to develop, as different adaptations begin to become more prevalent in the populations, and that ultimately will lead to evolution. Okay? So populations as a whole will evolve, not individuals. Okay? And so that's the first major misconception we're going to clear up. Okay? Individuals do not evolve, they just grow and develop, okay? But populations will grow and develop as a result of natural selection, kind of favoring certain adaptations that will ultimately lead to evolution. That's number one. So that's what we kind of saw with the finches that we looked at on Tuesday. So when we looked at the finches, the individual finch beaks didn't enlarge over that 2-year period after that drop. Okay? Their beaks didn't just grow in size, okay, from one individual finch. Okay? But what ended up happening was the big beaked finches were the ones that survived. Okay? Those big beaked finches mated with other big beaked finches, and they produced big beaked offspring. Okay? So the individuals themselves did not evolve big beaks, okay? But the offspring started to develop big beaks as we had these big genes being passed down from generation to generation. Okay, so that's number one. Number two is that evolution does not explain the origin of life. Okay, a lot of times one of the major misconceptions is that evolution explains how the Earth came to be, or how life, how life on Earth started, and things like that. Evolution does not attempt to explain how life originated on Earth. Okay? All evolution does is it attempts to explain how populations change over time and how we end up with life diversifying and producing new species. Okay? There were way too many factors that occurred during Earth's creation, and we can't recreate them in labs. So there's no way for us to know how life began. Okay, so evolution does not attempt to explain how life began. All it does is it attempts to explain the origin of species, okay, how populations change over time and how we ended up getting new species. It does not try to explain how the Earth was created or how life began. Okay. That's the second misconception here. Okay. The third misconception is that sometimes people would say that organisms do not evolve on purpose. Okay, or sorry, that organisms evolve in order to do something. Okay, organisms do not evolve on purpose. Okay, so a normal kind of common saying that sometimes people will come up with is that organisms will evolve in response to a change in their environment. Okay, well, that one we already saw that was false. Okay, organisms themselves are not going to evolve. Okay, other times people will say populations will evolve in response to a change in the environment. That is also false. Okay, populations cannot evolve in response to something. Okay, evolution is not intentional. It's not goal oriented. There's no kind of goal that is kind of accomplishing, that is going to get accomplished. And evolution is something that just happens. and it does not happen in response to a change in the environment. Okay? So evolution is not intentional. Okay, it's not something that you can actually try to do in order to help you survive better. Okay? So what will happen is the environments themselves will change, and as the environments change, that causes some of the individuals to survive better than others. Okay? So the ones that survive better are the ones that are going to be able to produce more offspring and pass on those particular traits or those favorable traits to their offspring, and they become more prevalent in the generations to come. Okay, so all the variation that is going to be present, that natural selection is going to work on, is already present in the population. Okay? So these beneficial traits don't just pop up in the generation or in the population. They're already present. They're already there. It's just a matter of whether they become more prevalent or not. Okay? So populations are not going to evolve in response to a change in the environment. Okay? Population is something that just happens. Okay? So here's an example. Okay, so an example of this is if we look at some bacteria, there's certain bacteria that are going to be resistant to antibiotics. Okay? So for example, here we've got a bunch of bacteria here. Okay? So we have a bunch of bacteria, and we're going to apply some antibiotics to them. Okay, however, there's going to be a handful of bacteria that are going to contain a mutation, okay, that are going to make them resistant to the antibiotic. They're going to be able to survive that antibiotic. Okay, so what we see here is that those without that mutation end up dying, okay, while the ones that survive, okay, are the ones that will be able to pass on their generation, okay, or pass on that mutation to the future generations. Okay. Okay, so we see a bunch of these guys dying here. We see some bacteria with that mutation that end up surviving. Okay? And as they survive, they continue to produce their, they are able to kind of pass on those genes to their offspring. Okay? Until you ultimately have a bunch of these individuals, a bunch of these bacteria with the mutation. Okay? So again, that mutation wasn't there to help them survive. The mutation was already present in the population. Okay? They did not evolve that mutation in response to the antibiotics. The mutation was already there. Okay? All that happened is that the mutation allowed them to be able to survive and reproduce, okay? And that mutation was then passed on from generation to generation until ultimately we saw the evolution of this particular bacteria. Okay? So this is part of the reason why it's not good to take antibiotics for long periods of time, because if you have any bacteria that are resistant, they're going to be able to continue to develop and develop and develop, okay, without dying, and you're basically just going to continue adding antibiotics that are not doing any good. Okay? So those mutations were already present in the population. Cool. Okay, so last slide here. Evolution, like we said, is not goal-oriented. Evolution does not attempt to make organisms better or stronger or faster or more complex or anything like that. All that happens here is that we have advantageous traits that are passed on, or that are passed on to generations, or passed on to their offspring. Okay? However, those advantageous traits, okay, may become disadvantageous later, okay, as the environment again changes. Okay? So evolution is not goal-oriented. Organisms do not evolve on purpose. Populations do not evolve on purpose. They just kind of happen. Cool. Okay, that is it for today. Okay. Any questions on anything we've gone over from chapter 11? Okay, so if there's no questions, you guys are all free to go. The homework is up on Canvas, the classwork is up on Canvas as well, so make sure you complete those. And then, yeah, so have a good weekend. Make sure you're studying for the final, and then I will see you guys on, if I see you guys for lab, I'll see you guys later. If not, I'll see you guys next week